Okay. Well, hey, everyone. It's my pleasure to welcome you to this joint webinar with Voxeleron and Voiant on how interoperability drives ophthalmic clinical trial success. Before we proceed, I'd just like to remind everyone there'll be a Q&A session at the end. So please submit your question via the Q&A feature, and we'll try to get to it. My name is Daniel Russakoff, and I'm the CTO of Voxeleron. On the Voxeleron side, we also have Jonathan Oakley, our Chief Scientific Officer. And we're also joined from the Voiant side by Hannah Rodriguez-Coleman, Voiant's Associate Medical Director for Ophthalmology Imaging, and Jason Slachter, the Voiant's Medical Director. And I will just want to do one more reminder to add your questions to the Q&A at the end. We'll try to get to all of them by the end. But first, I had one question for myself. It's for you, Hannah, and it's I thought you were with Dark. What's Voiant?
Hi, thank you for inviting me. Voiant is the rebranding of three industry-leading companies, MedQIA, WorldCare Clinical, and Dark. We've come together to form Voiant as a new leader in clinical trial imaging endpoint data. Our team, the Voiant team, has interpreted over 500 million images from thousands of global clinical trials. And we've aided in numerous biopharmaceutical companies, bringing new treatments to patients across ophthalmology, as well as oncology, respiratory, cardiovascular, gastrointestinal, and liver disease domains.
Cool. So, given your position then as the largest independent CRO, what critical technology gaps in ophthalmic image analysis have you encountered?
Well, in ophthalmology, one of our biggest hurdles comes from the multiplicity of manufacturers. We love our toys in ophthalmology, and each of these devices have developed different algorithms. In OCT, for example, for thickness analysis, some devices use ILM to RP, others ILM to Brooks. They also vary significantly in the quality of display capabilities. This multiplicity of presentation and of display has limited cross-comparison analysis in large clinical trials. Before we started using the Orion system from Voxeleron, sponsors couldn't really do a direct comparison of raw data across the different devices because there's no standardization. Instead, they had to rely on a relative percentage change from baseline for all of their analysis. This significantly limited the pool of sites that could participate in a given study, depending on the system selected by the sponsor and on the availability of each device at the site. As you know, when you limit the number and/or distribution of sites that can participate in a trial, you're limiting not just individual patient access to participation in a potentially groundbreaking therapy, but large population groups as well.
And then how does this lack of interoperability that you mentioned exacerbate the challenges you're facing here?
Well, from a reading center point of view, it makes it impractical to view these different images across different modalities. It limits the availability of site certification, or the probability of site certification, depending on the devices that they have, because of the limitation to or setup of the algorithms that you have to use by each device, it also limits the analysis that we can provide. With Voxeleron, that changed everything with Orion.
So it's fair to say it's easier on your readers to use one single piece of software.
It is easier, it is more effective, and we can provide more data in a more efficient manner. Better data in a more efficient manner.
Okay, yeah. So it's clear ophthalmology presents some pretty major challenges. So how have you and Voiant and the Voiant team overcome these challenges?
Well, because we are aligned in our intent to deliver the best quality data, working with the Voxeleron team in this common view and platform that is Orion, we are able to um, look at images from different devices, from Spectralis, from Cirrus, from Tacom, from a number of different devices in one common view and platform. We're able to work with the sponsor to apply the segmentation algorithm of their choice to develop the outcome of the data of their interest in an efficient and easy way.
Got it. So sounds like we have a pretty good idea, or a pretty good understanding of your solution to the problem. Why don't we walk through how you're using the Orion platform now?
Absolutely.
So it looks like this is not a view you're used to seeing because Orion has an API that can be called, but this in general, if you're using it independently, you would just open up a file here. This is a Spectralis E2E file. Once it's open, this is a view that you would be more used to seeing. These are just the B scans aligned and scrollable. If there's an LSO image, it's also displayed and registered. Just to highlight the interoperability, this is a Carl Zeiss Cirrus image that we're loading. You can view it in the same way.
And from our perspective, we would just be viewing it by study and by patient without real, without that kind of drop down.
Yeah. This is a TopCon FDA image. This is one with pathology, and this gets at another issue. It looks like the fovea is wrong here, centered at the center of the image, but the fovea is actually not there. Previously, what would you do in a case like this?
In various devices may have the ability to correct centration. Not all of them do, but it can be difficult to set up, difficult to reproduce. Depending on the pathology, depending on the cooperation of the patient, the scans can be limited, and also depending on the experience of the photographer. In the past, for the devices where centration was not possible, that was a lost data point, a value where the CST, the central subfield thickness, could not be calculated because the central subfield wasn't properly being captured or centered in the grid, so that the algorithm of the device couldn't be applied. The fact that in Orion we can correct it just by finding the umbo on the scan as we're viewing it and right-clicking on the thickness is easy, it's revolutionary, and it's exciting.
Well, and that's what we're doing here. And so it's resegmenting. That looked like it took about 2 or 3 seconds. Has that been your experience with the Orion software?
Absolutely. With you guys, recentration takes as quick as it takes me to find the umbo on the scans.
Great. And now, so you're
And
right.
as
Go ahead.
And what one of the really interesting features of your recentration correction is that in doing that, not only does it correct centration, but it also adjusts and corrects the segmentation to that new point, which is not something that the other devices will do. Those lines that blinked on and off, that's a recalculation of the ILM, of the RP, and all of the preset intraretinal layers that you have that happen in the same button.
Well, here's not the segmentation doesn't always work, and I guess you can't expect it to. So here's a
Right.
pathology
With
case where the layers aren't correct. Yeah, sorry, go ahead.
with any pathology, there is going to be, especially with significant pathology, it is expected to have some deviations. Part of that is because the quality of the image degrades, especially with retina thickness as it goes towards the deep. So it is harder for an algorithm to work. But with the steps that you're seeing, just by moving those dots, the ease through which it can be corrected with this intelligent editor, which goes through 7 or 8 individual lines, and then propagates quickly through the entire cube, is fantastic.
So yeah, so we're looking at the intelligent editor here, which takes a rather than going slice by slice, takes a series of slices through the cube for maximal coverage that you edit, and then,
Mhm.
as you said, propagates through. Can you speak to the ease of use of this versus other software for correction?
So there, the propagation technology that you have and the intelligent editing technology is very intuitive, which is not something that I have often come across. The ability to correct in the preset lines and also add your own for so that white diagonal line is one that can be manually added to oversample an area that is particularly of notable pathology. Um, I haven't seen that before.
And
And
here's
not only are you working with the presets, but the ability to add um, another sampling line is makes it so much quicker. I'm sorry, I interrupted you.
no. So here's a good example, though. So sometimes you don't need a full layer intelligent edit. Sometimes you just need to touch things up.
A little tweak.
Yep, a little tweak, and
Yeah.
that's what's sort of being demonstrated here. How often do you use this?
We use it a fair amount. The automated segmentation that happens when you first correct centration, most of the time is quite accurate. So our readers will go right into viewing the slices. We evaluate in the reading center every single scan in detail, and the being able to manually correct those slight blips as you're evaluating cuts down on time, and makes it much simpler, almost two birds with one stone type of effect, especially when the deviation is so minor.
I'm going to interrupt if you can hear me, and I hate to interrupt, but you made a statement a moment ago, Hannah, and I think it's very important to reiterate. Whenever we talk about adjusting the blips or resegmenting these things, the important thing is where you started, and the quality of the algorithm on the Orion program is so superior to the native software packages that come with the commercial manufacturers that the readers automatically start at a far better point. So there are fewer blips to correct, there are fewer errors to deal with, and then the system takes you even further in making it easy to correct it. And I think that's been one of the most exciting things about this whole process is every single step we talk about is a major improvement over everything we've had. When you add up those improvements, you've changed, as Hannah had pointed out, the accuracy and the efficiency of our readers, which is ultimately what sponsors, contracting reading center are looking.
Absolutely. I mean, the Orion segmentation algorithm is so far above other modalities, and adding the ability to fine tune that, it really ends up with next level quality data. The ability for us to deliver next level quality data to our sponsors.
No, that's great. So let's see, moving on to the next demo. This is so we've seen how Orion helps you capture the quantitative values used in a clinical trial. This is an example of recently we've introduced what we call the CRF panel, the case report form panel, that lets you capture the qualitative results typically associated with a clinical trial or with a case report form. So here, for example, we're indicating that there's the presence of epiretinal membrane, subretinal fluid, and PED. So basically, you can have both the qualitative and the quantitative information all in one piece of software, essentially the entire CRF.
I am looking forward to that.
Yeah. And we can also use this API to associate the quantitative measurements with labels. So like here, for example, the calipers are showing central retinal thickness. And it is important to note it's an API, so it's been defined and opened up so that each time you open Orion, you can see exactly the analytes that you want for that image, for that visit, and for that specific trial. Here, PED height. And this API populates it populates the annotation functions too. So you can, as you're adding text annotations, they are associated automatically with your qualitative measurements for examples and QA purposes. We haven't rolled this out yet for you, Hannah, as you mentioned, but is it safe to say this will, I guess, hopefully speed up your reads and improve transcription errors?
Yes, I mean, eliminating a transposition step is going to make the whole process more efficient and effective. Absolutely.
Great. So yeah,
I'm very much looking forward to it.
Yeah, we're looking forward to rolling it out to you. I think the next demo we're going to see relates to intraretinal segmentation.
So I'll talk a little bit to this and ask you about it, Hannah. But so this, normally the endpoints, I guess, are typically total retinal thickness, but that can obscure the fact that there's RPE elevation relative to Brooks. Here's an example of dry AMD where we are also quantifying drusen volume. So the average thicknesses between the RPE elevation and Brooks are reported within the ETDRS grid, and this obviously is a volumetric measurement as well, given a known area. Can you speak to how you use this in clinical trials?
Yes, so not just in RP elevation or drusen volume, but also we have more and more studies of inherited diseases, of glaucoma, that are looking at very specific intraretinal layers, thickness, and the ability to correct those layers, the RP and the Brooks, to get an accurate volume, drusen volume, the RNFL and IPL to get those thickness values for analysis, and the customization of the output to what the sponsor needs is something that we have been getting more requests from, and we're very excited to be able to provide with your system.
Right. So what we were showing there as well was the intraretinal layers of the ganglion cell complex from the ILM to the posterior of the IPL. And you can also peel away the RNFL and just show the posterior RNFL to the posterior of the IPL. That's the GCIPL layer. Some devices give you one thing, others give you another one. Orion gives you both. And it's showing here as well the ability to just change the image appearance. You can either do that automatically or manually. But extending the notion of having these additional layers that, as you said, are relevant to your clinical trials. I think there are situations where you might want to add a layer. Here we're looking at adding a layer to the choroid sclera interface. Can you talk a little bit about why you might want to do that in a trial?
When you have the ability to add a layer to specifically look at a thickness or an area that isn't being evaluated elsewhere, you're really providing sponsors with the ability to better understand outcomes of therapeutic intervention as applicable to the study in general, or even to specific populations of part of the study, and to be able to customize those planes of evaluation is really quite interesting and exciting, and applicable to many of our studies that we're already doing.
Excellent. So for those looking at this demo here, what we've used is the same intelligent editing mechanism for adding a layer. So rather than going slice by slice to define a surface that's continuous through the 3D volume, we've done these different views, as was described during the editing, to define that. And then that allows the algorithm to interpolate and cover the entire surface. So now we have choroidal thickness. And this is in a standard spectral domain image. If we hold here a second, so what's going on now, I'll talk through this, is we've loaded an E2E. The E2E files are typically multiple series. They come from the Spectralis device. And one of those in this case is fluorescein image. And this is a 2D segmentation algorithm that's semi-automated. So here we can see that the user has to define, so the first thing they did was just zoom in on the area of interest. And now they're just defining the regions of interest. So here in a geographic atrophy case, you can quite clearly see the hypofluorescent regions. And these are the ones that would then be segmented to give you an area measurement. And so the user interactions here define, well, what's the foreground, the measurement of interest versus the background, and then let the algorithm do the rest. So common to the tools that we have where there's a user always in the loop in the clinical trial scenario is that we maximize the measurements that are made, yet minimizing the user requirements, all the while maintaining accuracy of what's being reported. So what's happened now is the algorithm has performed a segmentation based on the user's input. And this is just with the sketching of these particular regions of interest. And as you can see now, the user wasn't completely happy with what had happened. So they're now iterating to a solution that they can report. The area is measured exactly per the metadata that is in the E2E. So we know exactly the fields of view, lateral fields of view in X and Y. And so these area measurements are then an accurate reporting of this particular 2D fluorescein angiography image.
That's fantastic. The ability to, with any system, there's always challenges and hurdles, but Voxeleron has repeatedly demonstrated a response of addressing those and in very innovative and intuitive ways. I like that.
Thank you. Yeah, that talks directly to this slide, accuracy, speed, and versatility. And so is that a fair summary of the benefits that you're seeing using the platform, that the time?
Absolutely.
Accuracy? You touched on variability. Because the algorithm component now is common across the different image formats.
Mm-hmm.
So variability is reduced as well. Is that fair to say?
Yes, access from the ability for different sites to participate in a study and still have the sponsor be able to do a point-to-point comparison, not just a change comparison, is revolutionary. It increases the speed at which a study can enroll by increasing the number of sites that can participate. The accuracy of the intelligent editor and of the process, it increases the speed in which our readers can generate the data and develop the data, and it's fun, fun to use.
So an overall impact, would that include higher completion rates to your trials?
Yes.
I mean, in a really broad perspective now.
Yes, I think that by being able to deliver relevant and important outputs to the sponsor, by the sponsor being able to select the algorithm of their choice, whether it's ILM to RPE or Brooks for thickness, or ILM to RNFL, or Drusen volume, RPE, all of those together, and to be able to do that using multiple devices is going to have in my opinion, a very big impact in the field.
So if I go off a bigger picture, what would you say is next for Voiant as you go forward now with these new tools? And
We
before we
are,
open up for questions in general, can you speak to what's coming? And
as I mentioned earlier, I think that the versatility of the tool to analyze the different layers of the retina increases the number of diseases that where it becomes relevant, that we can study it. So it's not just diabetes or just macular degeneration, but it is inherited diseases. It's glaucoma studies, it's safety studies for evaluation of toxicity in a particular layer, and the further, not just thickness or area, but volumetric analysis really will impact that the field in being able to tailor it to understand the outcomes of therapy, to better understand outcomes of therapeutics and populations in which it would work best.
and
I'm
then
just gonna,
sorry,
if
Jason.
it's okay to jump in, just
Yeah,
to,
sure.
to add to what Hannah said, you know, very often as a reading center / consulting team that we try to serve at Voiant, you know, sponsors will come to us. Some know their product well, some are very experienced, others are new in the area. They have a product, they're excited about it, they want to understand. And unfortunately, until recently, the discussion is, we would like to know this, but what are the limitations in imaging with regard to what can we learn about our product? And we're moving into an era with this type of software package where we can start to say, what do you want to know, not what can you learn? And this is really changing that paradigm, where if you've got a product that you want to understand the details of how it's acting, if you have a therapeutic that you would like to understand what area of the retina, what part of the retina is being affected by it. We have evidence that it's an outer retinal therapeutic, or an RPE therapeutic, or a drusen reducing therapeutic, whatever it is. We can now offer data to, you know, answer those questions. Whereas in the past it was, well, that would be great to know, but we can give you central subfield thickness kind of somewhat accurately, maybe not. And that's all changed now.
Yes.
That's awesome. So you're offering more and more advanced endpoints as well, it sounds like.
Yes.
And the endpoints they want and they
Yeah.
need, rather than what we can offer. Yes, we can offer anything they want essentially now.
That's awesome. All right, so we're going to go toward the Q&A session. So this is open to everyone to jump in on any questions related so far. We will start by seeding some questions, and first one's to Hannah, and I'd like to ask, how much faster, if at all, are your reads now with the Orion tool?
To the extent that we can provide more information and more data that we ever could. It is, you know, before, like Jason said, central subfield maybe, that's still 5 seconds, 10 seconds. But really the focus here is how much more we can provide on the analysis in a very efficient way that I couldn't tell you how long it took me before because I couldn't do it before.
Interesting. So
It is faster for the limited data points. I'd say it's about 20-25% faster. But really the point is how much more we can provide in the same time.
So you're doing things in roughly the same time, but you're providing more measurements, more accurate measurements. So the sponsor's gaining here,
Yes.
rather than, okay. So that's very
more relevant data is what we're able to provide, and more accurate.
I have a question in the session. Can you speak a bit to the setup process, how easy it was to get up and running?
From a reading center point of view, I mean, that is working with you guys, working with the Voxeleron team, it has been easy. from my perspective as a reader, it was really just pressing another app in my interface. But the responsiveness of the team and the depth of expertise has really been incredible.
So I will ask the next question. So you touched on one of the advantages that you have seen already from the change of interoperability, in that you worry less about what device is coming because you're going to deliver the same measurements regardless. Right. You don't have to worry about that. So you're effectively, are you saying that this is now a larger recruitment pool? And if so, has the kind of threshold on the, you know, sometimes you cannot grade, right? So if you're now saying that you've got a larger recruitment pool, so you have more, you're open to any device, and you can make a read on any of those devices, and you have tools to first automate and then quickly correct, how is the cannot grade threshold changed, if at all? So
so it's decreased it significantly, because we can correct one of the highest rate of cannot determine or cannot grade related directly to poor centration of a scan,
okay.
or incorrect segmentation for that central subfield. And one of the interesting things about Orion is that we receive the raw data, not the process data, and we're able to change the specifications to adjust it to better see a particular layer or a particular area in a customizable way, and still return to the raw data. So the number, while of course there's always going to be images that are subpar related to pathology, you know, if the cornea or the cataract of a particular patient is too thick, that the images, there's not much we can do. But those errors of centration, those errors of segmentation that come from the pathology that we can correct, is data that we can now use that we couldn't before.
So in some respect, your effective yield is
much higher. Yes.
Okay.
Much higher.
Just a reminder to get your questions in via the Q&A feature to have them answered. This one is for you also, Hannah. Is there a portal or a server to store and manage the data for this, for Orion?
So we have yes, there is. We use Orion through our independent servers, and we have access and control of that data as a reading center, which is different from your cloud interface, I believe, and the ability to set that up with our technical team. Well, it isn't something that I can personally speak to directly from experience. I know is there, and we do have full independent control of that data.
Got it. I guess, and I can follow up a little bit, as you alluded to. But right, it's certainly worth noting that the Orion interface is built, has a command line interface that can be called from other programs, which is how you use it. And right, we have a cloud-based clinical trial data management system as well, called iNebula, that also integrates with Orion. But you know, everyone has right, there's a lot of different ways to deal with the data, and those are just two examples. Let's see, I think that covers.
Just to jump in on that, I mean,
sure.
That as somebody that watched all the emails go back and forth and the phone calls internally, when we first added Orion system into our server structure, I was nervous. A few of us were nervous. I have to say that the efficiency with which the process was accomplished, you and the team's knowledge made it seem so much easier than I expected. I really have to say that we have our SOPs, we have our methods of operating where we maintain our own secure server systems, and the ease and adaptability of the Orion software package was nothing short of shocking to me, because I expected problems. Everyone knows whenever you even upgrade a software package on your computer, you look for problems. We were installing a whole new OCT evaluation package, and it went beautifully. And whenever we did run into a hiccup, you guys fixed it. So I don't want to let that go, because it was a major thing for us and well appreciated.
Oh, thank you.
Thank you.
Here's
a question that's come in. And so, Hannah, I know our answer. I'll give it in a second, but it says, are there any OEMs or protocols? So I think this means formats that you're not able to work with. And I assume imaging, so ophthalmic imaging.
So none of our preferred devices have had that problem. They're compatible with the Orion interface. As the reading center, quality of image and received is really important. So we do go through a certification process both for the devices and the photographers and technicians that take the images. And of the ones that pass our robust process of certification, those have all been compatible with your system.
Perfect. I mean, the answer from the Voxeleron side is no. There's none that we know of that we're not compatible with. So then if there's a new device out there doing some sort of imaging that we haven't heard of, then maybe. As of today, no, is the answer to that one.
Okay, well, I think that looking at it, I think this is their last chance to get questions in. I'll maybe wait 30 seconds for last question or two, if anyone has it. Otherwise, Jason was gonna ask you if you had any summary remarks, and we can adjourn. Looks like that.
I think that as I've said before, we have as a reading center is to deliver high quality, reproducible data to enable the people that hire us, sponsors and CROs, to get the data they need to be able to assess the safety and efficacy of a clinical product for the ophthalmology division of Voiant. That would be for treatments of eye disease or systemic therapies that have potential ocular toxicity. That's our goal and our mandate. And as Hannes started out by saying, one of the biggest limitations we faced was in OCT. OCT, as everyone on this webinar knows, as every practicing physician in ophthalmology knows, OCT is a mainstay of our evaluation process of different diseases. And we dealt with the growing number of manufacturers, a growing number of versions from manufacturers, and we had to incorporate all those. We had to bring all those in, and for several years, manage them individually and limit the data that we provided to the sponsors, which meant we weren't fulfilling our mandate. We weren't fulfilling our mission. And since we started to work with you guys, the Voxeleron team, and incorporated Orion, that has turned around completely. OCT is not looked on as anything other than a growth and expansion area. It's something that we can generate new, exciting data from as we start to dive more in more detail in the different layers and analyses. And that's been entirely due to Orion and your team. And I just have to say that it has been nothing but a pleasure. We set upon trying to find a partner when we looked upon going to a common platform system. We looked at various companies, we looked at various groups, and we were impressed with you when we started the process, and we've grown to be more impressed with the Voxeleron team and the capabilities of the Orion software as we work together. And I'm looking forward to many, many more years of collaboration and our ability to fulfill our mission.
As are we, and
Yes, that's
that's
Hear,
it's a
hear.
great. It's, I was gonna say, it's a great way to wrap up, but I apologize because the question just came in while you were talking. How has this assisted in looking at OCTA or OCT angiography images?
So we currently are not looking at OCTA with the Voxeleron platform. It is something, one of the things that we have in our list to do, but we have not started that process.
So I can jump in and help talk about how Orion supports that. So again, we support all the major devices, all the OCTA data that we've seen come in, we support. I say the first primary thing is with the Orion and OCTA is the definition of the plexuses. So because we have this excellent layer segmentation and the ability to change the boundaries very quickly, then we are able to clearly define the plexus of interest. So be it from the inner superficial to the choriocapillaris, they're very well defined. And as you've seen, you can very quickly add layers if you want to make a particular band of definition. Then the measurements that we automate in there are the fovea avascular zone. There's 2 algorithms in there that you can choose from. One internally developed and 2, one that follows the literature. And so those are like anything we automate, we edit tools so that you can change those measurements. The other one we have is capillary density measurements. So when we were coming out with OCT analytics, it's still it's not clear where everybody's landing. There was thoughts on you show the vascular densities and so on, or you show avascular zones. So kind of 2 sides to same coin in some way. So we have capillary density measurements that's following if someone sends me I can send the paper that it follows, but we don't have the areas of non-perfusion. Sorry. So we have capillary densities and not areas of non-perfusion. But there's the freehand drawing, the annotations. There's a lot of measurements that can be made automatically. You can measure lesion size and so on that you see very clearly defined by the plexuses, again, by the layer segmentation. So it's an area that we intend to develop more and more algorithms for, but there's limited consensus in what to use there. But as Jason has so eloquently said, we do work with people to develop endpoints as required. And we have done that because a lot of the tools that you've seen are actually driven by Voiant, because people like Hannah are experts in these areas and say, well, wouldn't it be great if you could do this instead and do it this way? We're not the experts in workflows or the measurements to be made, but we can implement these when we're guided correctly by the experts in these areas. So where we're lacking in OCTA, we can certainly make up, but right now we follow some of the standard measurements in the literature, but again, have at least a common algorithm doing the measurements that are there. And secondly, the user has the ability to override any of these results that are given. I hope that helps, but if you follow up with me separately, I can I can send you some literature.
Yeah, it's exactly that responsiveness and that willingness to work and on my "what about this" statements with no pushback has been one of my favorite things of working together.
Oh, thank you.
Well, I'll just throw in very briefly. It's worth noting we support all the OCTA formats from all the main devices. I think that hopefully that goes without saying based on our reputation and what we've mentioned, but I'll say it anyway. Okay, well that give it 10 more seconds for a final question. I don't see anyone coming. I think that's it. So just want to thank you all for joining today's webinar. We'd like to also say thank you to Voiant and Hannah and Jason for co-hosting this event with us.
Thank you.
If you'd like more information or would like to request a demo of Orion, please visit our website at voxeleron.com/Orion. Thank you very much. It's been a pleasure.
Thank you.
It's been a pleasure. Thank you.
Bye.
Thank you very much.